Just a quick introduction. I'll talk about the Molecular and Translational Imaging Core, which I am part of, and then preclinical imaging at CAMI, the Center for Advanced Molecular Imaging. And then we'll get into the meat of the talk, which is what is MRI and how can you use it for your cancer research? And then, as the title suggests, there's an alphabet soup. So I'll talk about these different types of MRI, and then I'll round it out with some cell tracking experiment or examples and some quantitative imaging. So the Molecular and Translational Imaging Core covers both campuses. On the Chicago campus, you have the Center for Translational Imaging that's in the Olson building, and we are in Silverman Hall in Evanston. Both cores have a 7 Tesla MRI, and CAMI also has a 9.4 Tesla MRI. We have INIVIS for optical imaging, and both facilities have a Mediso PET CT for nuclear imaging, and CAMI also has a SPECT imaging. And I'm going to focus on MRI. If you're interested in some of these other modalities, you're welcome to reach out to me. And then all the clinical imaging is done through CTI. They also recently acquired a PET MRI. And we have support facilities in Evanston for our cell culture room and our own vivarium run by the Center for Comparative Medicine. We offer advanced image analysis and visualization. And one of the things that's unique to CAMI is we have all of this equipment and infrastructure in one location. Was designed on purpose to have the vivarium here and the MRI and IVUS all within like eyesight of each other. So we can do multimodality imaging. So you're probably wondering, if you're on the Chicago campus, how can you work with CAMI? Well, there's a van that transports, it's not that van, but there's a van that transports animals between campuses. It's all run through CCM, so there's no issue of quarantine or importing. It's all considered within the same university. So it's pretty seamless. You just, if you're familiar with AOPS, you put in an AOPS transfer to another room, and then behind the scenes, they understand that if it's a room in Evanston, it requires the van. There's free transportation on Tuesdays and Thursdays. So as I mentioned with the floor plan, we have pretty much every preclinical imaging equipment. So if you can imagine what you would see in a hospital, we have the small animal version. The only thing we don't have is ultrasound. Just to orient you as far as scale, so we are in this region here, which is about 100 micron, is as small as we can go. And then for comparison of scale versus sensitivity, you can see on the slide there that they're essentially inverted, with micro CT having the highest spatial resolution but the lowest sensitivity. And then MRI is kind of in between, which is what we'll be talking about today. For preclinical imaging, you can essentially divide by 10. You get about 100 micron for MRI and say 30 to 40 micron with micro CT on a preclinical scanner. So a lot of times people mix up MRI and CT. MRI is magnetic resonance imaging. CT is computed tomography. So just very quickly, what is CT? So CT is a series of X-rays that are taken as the gantry rotates around the sample. So you typically have an X-ray tube on one side and a camera on the other side, and it rotates around and it takes a 2D image or a 2D X-ray, and those are referred to as projections. As you increase the number of projections, you get a better sense of what the object was that attenuated the X-ray. So this is an example of what the, let me get my laser pointer back. So this is an example of what the reconstruction looks like as you increase the number of projections. And then this is what the raw data looks like. This is a monkey skull that I got from the Field Museum. It doesn't have anything to do with cancer, but it's kind of neat. So this is a series of projections taken with the camera going around the skull, and that's what the raw data looks like, and you use an algorithm to turn that into a 3D structure. One of the important things about the difference between CT and MRI is CT, especially on a preclinical scanner, doesn't have very good soft tissue contrast. So you can see here, this is a CT with SPECT overlain, which is a type of nuclear imaging. And this project, they were trying to develop a radio tracer to go to the pancreas. And so the person I was working with said, well, this structure here looks like the pancreas, so looks like we did it. This is the same rat with MRI. And you can see this structure here is the spleen. And you can see when you overlay them, their agent went to the spleen, not the pancreas. So soft tissue contrast is much better without doing any tricks in MRI. Anyone have an idea what this is? This is my litmus test to see if there's any MRI people in the audience, so I know I have to be more careful what I say. This is case-based data. This is what the raw MRI data looks like. So as it happens to be a data set for 3 slices, and as you go across, these are multiple echoes. So how does MRI work? What we typically image are protons. There's other things that you can image, which I've got listed down at the bottom there, fluorine, for example, or sodium. But most people image protons, and the proton interaction with, say, tissue, bone, air, fat, all those things contribute to signal or contribute to what we see in the image. So the first thing I'll mention is the spin lattice, which is called T1 for relaxation time. And that measures the time that it takes for the proton to realign with the main magnetic field. And I have a cartoon that will hopefully run so you can see that and it'll make more sense. And the second type of relaxation time is T2, which is spin-spin relaxation or transverse. So what you can imagine, if you have a top spinning, when you perturb it, the spin lattice is when it goes back to perfectly align with the magnetic field. And the spin-spin is when you tip it over and then you make it go back. And then T2 star is a little bit more complicated. That's where it interacts with local magnetic inhomogeneity. So I'm not going to get into that. So now hopefully this works. Let's see. I will try to fix that. Okay, so with this cartoon, you have molecules in a magnetic field. Turn on the main magnetic field, they stop spinning. You send a radio pulse, and you see how some of them interact with the radio pulse. That's how you get an MRI signal. And for the T2, the second type of relaxation I mentioned, it's when you invert them and then you measure the time that they invert back. So hopefully, let's go back to the presentation. Okay, we're back in business. Sorry for the delay. So it's lunchtime. Here's your alphabet soup, as promised. So these are the main, there's a lot of different types of MRI. These are the main ones that I'm going to focus on. Diffusion weighted imaging is when you measure the water interaction with the tissue, for example. So you can imagine as water travels through muscle, it'll have a different diffusion than if it goes through, say, a solid tumor. And those differences in the water can be turned into an image for diffusion weighted imaging. And it's quantitative too. So for example, you can get an apparent diffusion coefficient and get what's called a functional image and not just the anatomic image. Related to that is diffusion tensor imaging. So you add directionality. So it's not just the diffusion of water, but it's the direction that it's going. And that's useful in things like tractography for neurons, for example. Arterial spin labeling is a type of angiography. I won't get into that very much. Chemical shift imaging, where you interact with a contrast, typically with a contrast agent that shifts the frequency that the proton normally is at, which is field dependent, and you can do different things like measure pH or measure temperature with chemical shift imaging. I'll spend quite a bit of time on dynamic contrast enhanced MRI, which is my area of expertise. That's where you have a contrast agent, you inject it, and you measure the contrast agent concentration over time. It's typically a gadolinium-based contrast agent. Dynamic susceptibility enhanced MRI is similar to dynamic contrast enhanced MRI, but it uses susceptibility. So it's typically something like an iron oxide agent. In general, DCE MRI is bright as the contrast agent arrives, and generally DSE is dark as the contrast agent arrives. And you've probably heard the term fMRI quite a bit. That's a specific type, or it's usually referring to a specific type of MRI, which is BOLD, which stands for blood oxygen level dependent MRI. fMRI actually mean, functional MRI means anything that's functional, but a lot of times people use it interchangeably when they mean BOLD. And the last thing I'll talk about is echoplanar spectroscopic imaging, EPSI, which is very similar to BOLD, and I'll get into that in a minute. So those are the ones that I'm going to talk about. So in conventional MRI, you record this one point on the free induction decay. So remember, I was showing in the cartoon when you have the magnetic field and you pulse it with a radio frequency. As the spins go back to equilibrium, this is called the free induction decay, and it's dependent on the echo time. So conventional MRI, you collect just one point. With EPSI, you collect all of these, and that allows you to get a spectrum for each voxel and not just this single data point. So if you've never seen a pulse diagram before, this is what it looks like. So you've got, or I should back up and say that a lot of MRI is two-dimensional in the sense that you have X and Y and then multiple slices. So the third dimension is the slice. So you have a gradient for X and Y, and then this is the slice selection. So with EPSI, we add for each slice, the third dimension is time, and that's this red line here. So we acquire, if we go back here, this is acquiring the free induction decay in the third dimension. So why is that important to you? Probably not very important. But this is just an example of the different spectra that we can collect. And what we can do is we can do different tricks with the spectra to get Fourier components, which is a fancy way of saying these different shoulders, or if there's actually another peak that's resolved in the spectrum. And you can turn those into new functional images, and that'll make more sense in a minute. So these are just different examples. So this image down here is a rat leg with a tumor at the top. It's not a different slice. It's different Fourier components. And that difference in contrast is because you're focusing on different points of this spectrum. So you can see the little red dot there. So this is a better example of how you can take advantage of the spectroscopy with EPSI. Again, there's a tumor at the top there. This is muscle, and you can see how the spectra look different depending on which voxel you look at. And then you can actually do some analysis with asymmetry. So if as the name implies, if one side of the spectrum is not symmetric with the other, you can play some tricks, mathematical tricks, to get more quantitative information. And just this image is a functional image of that asymmetry, showing that the tumor rim is different than the tumor center, for example. One more example with EPSI. This is using super paramagnetic iron oxide contrast agent that we injected to look at tumor heterogeneity and angiography. Specifically, we were looking for microvessel density. So on the top is what's called a peak height image, which is just the peak of the water spectrum. And the tumor is at the top and the muscle is below. And then the kind of orange colored ones are the same slices that are a subtraction image before and after we injected the iron oxide particles. And the main thing that the kind of the take home message is these dark and bright spots are where the microvessels are, and they can be dark or bright depending on which side of the spectrum they enhance. So it's a little bit odd, but it's just the way that the spectroscopy works. So with a lot of imaging, you need to know ground truth, and a lot of times ground truth is histology. In this case, we used a barium cast. So this is a terminal procedure with the rats. We injected a barium gel, very similar to what you would get if you had, say, a CT of your colon. They'll make you drink this barium drink. So it's very similar to that. It'll coat the intestines. And it looks like bone, but all of these are large blood vessels. And what we're looking for are the red and blue from EPSI, which is overlaid, are they aligned with these blood vessels? And unfortunately, it doesn't show that well on the screen, but it did end up showing kind of overlapped. Come on. Well, that's unfortunate. It was working earlier. Maybe it doesn't like working through Zoom. So this is angiography. This is actually the neck of a mouse. And these are the jugular veins. And unfortunately, it doesn't want to play. But we'll move on. So diffusion tensor imaging, as I mentioned earlier, it's diffusion weighted imaging, but with directionality. So you can see this cartoon up here shows the directionality, and this is an example with like a fictitious neuron or something like that, and you can see how the direction would be influenced by the kind of the confines of the cylinder to reflect in the image. What you can get out of the DTI is fractional anisotropy, which is this FA here, axial diffusivity and radial diffusivity. And this color map here is where each voxel is color coded with the direction that the tensor is going. So things that are the same color are going the same direction. And this is a spinal cord example, which makes more sense than doing something with a tumor. You wouldn't imagine a whole lot of directionality with something like a tumor. So this was in collaboration with some colleagues at Medical College of Wisconsin. So this down here gives you a better idea of how the directionality could be picked up with DTI. This is a spinal cord injury where this is the injured axon right there. And that's what the image looks like. So because these are long scans and they're sensitive to motion and things like that, our colleagues developed a new version that has spectroscopy built in. So just the DTI alone didn't correlate with BBB, which is their behavioral training or behavioral measurement. But when you added the spectroscopy, it did. Okay, on to my favorite topic, DCE MRI. This work was done on a 4.7T, so this was done at University of Chicago. The main thing I'll point out is, don't worry, you don't have to look at the math equation. The main thing is this K-trans is essentially, or I should say, it's related to the slope of the gadolinium versus time curve. And this V sub B is related to the washout, and that will make sense when I show you one of the curves. A colleague of mine developed this empirical model, which is this one down below. The equation on the top is just converting the signal intensity to gadolinium concentration based on the T1 measurement. This empirical formula works really well, but we had a hard time figuring out how to translate any of these features or any of these parameters to something physiological. So if we go back to this equation, the K-trans can be described as perfusion, and the V sub B can be described as permeability. Those two parameters or physiological descriptions make sense, but when you go to this empirical model, we had a hard time combining parameters to get something as simple as perfusion or permeability. So these curves are what you get with the dynamic contrast MRI. And the main thing I'll point out is even without any modeling, you can look at these and notice that the slope is different between them, and also the washout is different between them. So what the modeling does is turn those into numbers so that you can get functional maps and also quantitative results. So here's an example with a colorectal xenograph using sorafenib, which is a kinase inhibitor. These are the T2 weighted images on the top. The bright thing is the tumor, the muscle is below, and the really dark thing is the bone. And the second row is the map of K-trans, which is one of the parameters that comes out of the two-compartment model, and then V sub B is the other parameter. And we did the analysis trying to, so I guess I should back up and say that the signal that these traces are synthetic. There's no noise. These aren't the real data. The real data has a lot of noise on them, and you're trying to model that shape with the noise built in. So one of the things we tried was take the region of interest, say tumor, and try to fit that data after averaging it together. And it turns out that it doesn't, the differences get washed out and the statistics don't work. So you have to switch to something that's pixel by pixel, which means for each pixel, you're going to fit that curve, even if it has noise in it. And in doing that, we found differences in the tumor day 0 compared to day 4 with the sorafenib and not with the vehicle. For both the vehicle and sorafenib, the V sub E didn't show any significance. And that really didn't bother us in this model. It's not as useful of a parameter, the washout or permeability parameter. So one of the next step that I was working on is, instead of trying to lump together things based on region of interest, like all the tumor or all the muscle, you've probably heard a lot about artificial intelligence or machine learning. Those rely on data sets for learning. And so I wanted to try something that isn't influenced by the learning data set or the training data set. So this is an example with fuzzy C-means clustering. It just says, if this curve looks similar to another one, I'm going to put it in the same cluster. That's all it does. So this was done in Evanston on our 9.4T Brooker with a gadolinium-based contrast agent. So this is a rat model of preeclampsia. You can see these are the placenta, and the fetus is here. And then we segmented and color-coded the placentas. That's what we were interested in. So the first thing we looked at is volume, and that's why we needed to segment them. You can see them rendered in 3D. I should say, as a side note, for tumor imaging or just tumor research in general, volume is a bad measurement. With MRI, you could see, for example, that the tumor gets bigger, but the interior is necrotic or disrupted. So just because the volume got bigger, does that mean it failed? Probably not. You could also see examples where the tumor volume shrinks, but the vasculature hasn't changed. So did it work or not? It's hard to say. So if the only thing you take home from today outside of the alphabet soup is tumor volume measurements are bad, especially if you have MRI available. So this is an example of these curves that I was talking about earlier. The Suramin is a antivascular agent, so it's the model is mimicking preeclampsia, and of course PBS is just plain phosphate buffered saline, and this blue and cyan color is just a region of interest in the muscle. And I've highlighted one placenta in each rat, and you can see the various curves that are color-coded. I like to call this the thin mint. So this little green line here is matched to this green line here, and you can see how it increases sharply. And then here on the Suramin rat, you barely can see that green line. It has a similar profile, but there's less of it. So what does that mean? It just means that the blood supply to the fetus has been disrupted dramatically in this preeclampsia model, which makes sense. So again, the light green is closest to the fetus, and this rust color is more of in what's called the labyrinth. And I just trying to point out that sometimes the shape can be the same, but the amplitude is different, or sometimes both the shape and the amplitude are different. So getting into some of the quantification, let's see, unfortunately, we have the zoom bar over the top there. This is just initial area under the curve. So if you remember, do we have a mark? We don't have a marker. Well, I'll just do it in here. Remember, the curve goes like this. So the area under the curve, you can pick different time points. And what a lot of people do is what's called the initial area under the curve. And you pick something like, say, the first minute, and that will get that peak, and you don't worry about the washout part. So without doing any modeling, you can just, using math, take that area under the curve, and you can see differences. So in this case, you see on, I believe this is day 18, you see a big spike in the normal rat that you don't see in the Suramin treated rat. And this is just the, how can I say, it's the rat getting ready for delivery. So all this blood going to the fetus just before the delivery. And that's attenuated in the preeclampsia model. And again, this is without doing any modeling. And the reason I bring up the modeling is because the modeling is not only influenced by noise, but unfortunately, different groups have different assumptions, different algorithms. So it's not really standardized. And that's probably the only downside of dynamic contrast enhanced MRIs. They're working on that, at the main conference. So at the International Society of Magnetic Resonance in Medicine, there is a study section that's working on standardization, but there's still a ways to go. So in this case, this is a preliminary study. There's N of 3. We didn't see any differences in K-trans or vis-a-vis. But one thing that we noticed that was interesting is the time that you measured, like when you start seeing the spike, the gadolinium contrast concentration increase, that's what you call time equal to 0. And that shifted in the muscle between the two groups. And we weren't really sure what was going on there. And we need to do a little bit more work there. But there's a clear difference between the preeclampsia model and the normal rats as far as that time to delivery. So this negative number here means that the placenta arrival time is delayed, and the positive number means that the gadolinium arrives at the muscle before it gets to the placenta. So it's kind of flip-flop between the two groups. I mentioned earlier that you need to know what your ground truth is. Typically, that's histology. And I brought this slide up just to point out that sometimes you can do some tricks to get better segmentation. In this case, you're probably familiar with RGB, red, green, blue color map, or sorry, color space. This is a less common color space called lab. And when you take this CD31 staining and use the lab color space, you can see these red dots are exactly where the staining is. So I thought that was kind of neat. One of the downsides with histology is there's tearing and stretching, shrinking, the process of preparing the tissue, especially if you do something like paraffin embedding. So this is just an example to show you how much warping is required to get something like an MRI scan to match the histology. So this is now, I told you they do all the clinical work at CTI. This is an example from University of Chicago when I was there, where this is human prostate. This is, we decided to use the ex vivo prostate in our 9.4T because of the difference in resolution. We were trying to look for smaller features that you wouldn't be able to see with a clinical scanner. So a clinical scanner is typically on the order of, say, 1 millimeter, half millimeter, something like that, whereas the preclinical scanner can do, 100 micron resolution. So this is the 9.4T scan. This is the histological scan. This is diffusion weighted MRI, and this is just a T2 measurement of that same ex vivo slice. The other point with this slide is sometimes you just get lucky. There's no warping here, and you can see this feature here is basically represented in all the different scans. By the way, the tumor is at the bottom here, which you can see this really dark T2. But this example where you have the in vivo MRI, the ex vivo MRI, and then the fused, the combination, they don't match. So this curve here is from the endorectal coil, and I'll show you a 3D rendering where you can see the bladder pushes down on the prostate. So that makes, hopefully we don't get stuck again. All of these videos were working earlier. I wonder if it's the combination with Zoom. Oh, here we go. Now the question, is it going to run? Well, even if it doesn't run, it's okay. You can see the bottom of the prostate is sloped compared to the ex vivo one. That's from the endorectal coil, and then it's sloped up here from the bladder pushing down. Let's not worry about the video. So the in vivo scan suffers from poor sampling, mainly due to the slice thickness and resolution. And then the time between the in vivo scan and the ex vivo scan, at least in this project, was often months because you're doing it the same day that they're getting their prostatectomy. So it's usually not the same day you get your MRI scan. So you get your MRI scan, you get your biopsy, and then they decide, are you a candidate for prostatectomy? And then they were enrolled in this project. So the gap between those mean that also the tumor and the prostate could be wildly different from when we actually put it in the scanner for the ex vivo imaging. Let's see. I already mentioned that. And this is an example of, the direction that you go for registration plays a role, whatever the final target is. So if you want to get everything related to ex vivo, it has to be in that resolution. So if something is low resolution and you register it to something higher, you end up, it's undersampled. So you kind of have to make things up, which makes more sense in this example. You can see these artifacts down here at the bottom because you're trying to create data where there isn't any. Let's move on. All right, so some examples with contrast agents. This is an iron oxide particle that was developed in Professor Mead's lab in collaboration with Jennifer Koblinski. She's at Virginia Commonwealth now. We labeled breast cancer cells with this iron oxide, and then they're also modified for bioluminescence, which you can see here. And it's a cardiac injection, and it just shows the metastasize to the brain here, which is what this model was. And hopefully this video plays. Maybe I have to turn the pointer off. No, it doesn't want to play. Well, we'll move on. I know all of the videos played earlier. This is a similar project, cell tracking with gadolinium-based nano diamond construct, also from Professor Mead's lab. There was actually enough contrast agent in there to have this dark image when it should be light, but we used a T1 measurement to show that it's actually in the left side, not the right side. So the same tumor on both sides, one was with the label and one was without the label. And this is just a cartoon, oh, not a cartoon, this is a phantom showing how the T1 measurement works. So the last part with alphabet soup, I already talked about, hold on, let me get my pointer back. So K-trans you'll recognize from DCE MRI. This was part of a workshop I did in Colorado where we were talking about different imaging endpoints. ADC is from DWI, SUV is from PET, but the last thing I'll mention, I think this is, this is the last thing. Texture analysis or radiomics, there's a lot of omics. So radiologists didn't want to be left out. So radiomics is, or actually texture analysis is a subset of radiomics, but it's just doing different mathematical tricks and turning them into images to get more information out of the data. So it's more than, how can I say, it's looking at different spatial statistics that's it's kind of hard to just see from the image. Hopefully that makes sense. Last thing I'll mention is I would be remiss if I didn't mention our colleagues at the Center for Developmental Therapeutics, or the Developmental Therapeutics Core. They work with us quite a bit on these cancer models. This is an old example with Professor David with an iron oxide particle that they developed. The other core that I'll mention is we're working with Quantitative Data Sciences Core. If you haven't seen this cartoon before, the gist of it is that the green jelly bean was significant, not any of the other colored ones. I recommend you take a look if you haven't seen this one before. But anyways, the statistics are important. So we're working with QDisc to develop a pipeline where we work hand in hand so that they understand the data they're getting, and also we work with the users to help them design their studies so that they have the right statistics. So a lot of this work wasn't done by me. I'll thank all of my collaborators. And the last thing I wanted to say is I was hoping to get this t-shirt to wear with the talk. If any of you are familiar with baseball in Chicago, this is Liam Hendricks. He was recently diagnosed with non-Hodgkin's lymphoma. And they actually set up something on their website where fans can send messages to Liam. So I was hoping to have the t-shirt so I could send a shout out to him, but it didn't work out. But anyways, I just think it's neat that they're, allowing fans to interact with him and sending him support. I should mention that non-Hodgkin's lymphoma is probably not a good candidate for MRI unless it's one of the late stages, and that's usually when you see metastases and things like that. So that's my talk. I am interested in cancer outside of what I do with research. So I also brought a prop. This is Hockey Fights Cancer. So thank you. If you have any questions. Hopefully there's a few minutes. Sure.
Or people who are getting into this and interested in, like, if there is a table available, then other resources that might say, like you said, there are different contract agents with these specific things. If you're starting out, you might not know what any of those are.
Sure.
There are these things, here what their best for detecting, and then
absolutely. Yeah. I forgot to put the reference for this. This was from a workshop we did, and we actually turned it into a white paper. So I forgot about that. So I can send that to you and you can disseminate it. So yeah. 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 And so if you see something on the list that's more related to what you do and it's not in my talk, I can talk to you about, for example, if you want SUV from PET imaging, I'm happy to talk about those other modalities too. Sure, go ahead. Absolutely, yeah. So if you can't do an IV injection, then the DC doesn't work, both in patients and in animals. So there are some studies that I've done where you do, 3 days after treatment and 7 days after. So if you start getting too many of those time points and you're not good at the injections, you don't have much of a tail left. So yes, you need to be able to do an IV injection. Well, that's how can I say, that's a good related question, which is for a lot of what we do, our users focus on either the therapy or the mouse model. So for example, we're starting a project with Tom Kume. He's interested in a genetic model for angiography. And if you knock out this gene, it's FOX something or rather, the angiogenesis changes. That's all he's doing. So what we do is we take his knockout model or mouse, and then we do DCE MRI, or we're planning to do DCE MRI. So all he does is focus on the mouse, and all we do is focusing on the image. So in your case, whether you're doing a therapy or a mouse model, you just stick with that. Don't worry about learning to inject, because you're also not going to learn how to run the MRI. We'll do that for you. If it's a big part of your project, then we do have a model where we'll train you how to run the scanner. And we found over time that people tend to collect garbage data because they do it after we train them. Then 3 months later, like, what's that one thing I'm supposed to do? And just let us do it for you, and it'll be a good collaboration. Sure, go ahead. Mm-hmm. Mm-hmm. There's still, so you can still perturb the water and get a sense of the movement of water. You're right, it's not the same thing ex vivo, but water, there's still diffusion. in samples. So you can do DWI on a sample. Doesn't even have to be a biological sample. Of course, it's less meaningful ex vivo than in vivo.
In this table,
So yeah, some of them require a contrast agent and some of them don't. Yeah.
proton spin, are there like features that intensify MRI?
Yeah, so going back to that slide where we're talking about relaxation times, that's without contrast agents. So the main thing you do with the contrast agents is look at the response to the agent. It's kind of misleading. You rarely are imaging the agent itself. You're imaging the effect it has on protons. So yeah, but we do have some targeted agents. So in a sense, you're imaging those. Sure. Just go to cami@northwestern.edu. You can find my email there, or chad.haney@northwestern.edu. And I think we're listed on the cancer website too.
Is some of the ones looking?
Mhm. Yeah.
Thanks so much, Becker.
Yeah, thank you.